So last time, most of the medical applications of safety and ethics we have talked. Like we talked about medical termination of pregnancy, euthanasia, organ transplantation. There is one thing which is there, which I added to your IVF, that is in vitro fertilization and cloning. That is another thing which I wanted to add here, because cloning is another very important, or stem cell research is another very important thing which involves a lot of ethical issues. Okay, so that is the other thing. So after this cloning and stem cell research, all the physician related where ethical issues are involved, that is done. Okay, most of the ethical issues which involve which are required, or which are in the medicine field, they will be done. For example, last time we did organ transplantation, euthanasia, and this will be the last. And this stem cell research is something which involves a researcher, where I am a researcher, not doing any medicine work, as well as a medical professional. Why? Because from where the stem cells will come? The stem cells will come from any medicine application. Either it can be an in vitro fertilization procedure where many embryos are frozen and then stem cell. So there is then those cryopreserved embryos can be used for stem cell. So stem cell research is one which is not other procedures which we did last time, like organ transplantation and euthanasia, medical termination of pregnancy. Those are exclusive physician based issues. One issue which is stem cell research or stem cell based work, or that involves both
Ma'am, screen
medicine
is not visible.
professional as well as the researcher. So this is what most of the time stem cell, whenever we talk about stem cell research, what are the ethics involved? Ethics involved is that once there are two types, how two ways how a stem cells can be used on a cloning purposes. Okay, for example, if I go back, if you see this is one very important. Many of you might be knowing cloning was done for dollies. So the sheep cloning, and then there are many animals where cloning is involved. But still, there is no till date human cloning is not allowed. Why? Because only experimental, which is called therapeutic cloning, that is allowed. What do you mean by therapeutic cloning? Means as the name suggests, that you have eggs from a woman, which will act as a voluntary donor. You have cells from adult tissue, which will be 2N, and you fuse them. You remove the unfertilized egg from a female, and you remove the nuclei from that egg, and then add those. It can be a skin cell. For example, I take out skin cell from one person, and you take out those somatic. So it's not germ cell. Whenever we talk about egg, sperm, or ovum, they are germ cells. But now what we are doing, we are adding the somatic cell to the egg. Okay. So what is the advantage? The advantage is that somatic cell is already a 2N. That is a diploid. And whereas whenever we talk about gametes, which can be ovaries, which can be ovum or sperm, they are haploid in nature. So after fertilization, it becomes diploid. Whereas in a normal, when we are talking about cloning, what we do, the environment of egg is stored, the nuclei is removed, and then instead of nuclei, we add the diploid somatic cells. Okay, because this is known as electrofusion. How you can add? Because there is a procedure where you can make a hole, electroporation, and enter that somatic cell into the egg. So that is one. Once, because this egg has this property of dividing, because it's an egg, because it's an egg, so it has cytoplasmic components which will keep on dividing. And once these cell division, and we have to give a lot of artificial environment to that. Once this is embryo. Why it is embryo? Because it is already a 2N. Okay, it will start dividing. It will start dividing. When it starts dividing, there will be a lot of stem cells. There will be there. So this is a blastula stage. This is the time when a lot of stem cells, it's full of this blastula stage, when it is full of stem cells, we take out the cells and use them, culture them, and use them for that same person from where this this somatic cell was taken out. For example, this first person from where we have taken the skin cell has Alzheimer's or has some disease, terminal disease. So once I can produce these embryonic stem cells, these stem cells can go back because it will be compatible to this person, and I can, because these stem cells are totipotent, they can be converted into any type of cell. So this person can be treated. So this is known as therapeutic cloning. So therapeutic cloning is allowed, whereas if you implant this embryo into a foster mother, that is reproductive cloning. Okay, so this reproductive cloning is not allowed because this is unethical. This is where ethics comes. Okay, so therapeutic cloning is allowed where you are not implanting the whole embryo into a mother or into a foster mother, and that will give rise to another organism. Whereas using that embryo for embryonic stem cells, that is allowed. Okay, so these are two things which are very important. That is known as, whenever we talk about cloning, therapeutic cloning is allowed, but not the. So most of the time, this is what, this is, as I was telling you, there would be many diseases, for example, cancer, Alzheimer's, Parkinson's disease. So there are many diseases where it's very difficult because the cells of the brain, which cannot get repaired, cells of the heart which cannot, cardiomyopathy, where cells cannot get repaired. In those cases, the stem cell replacement would be great. So that's why it's allowed the stem cell, embryonic stem cells, we are using them for therapeutic uses, that is fine. But still, there would be there are a lot of controversies, there are a lot of debate which is allowed, because most of the time people say that people who oppose this, even the stem cell debate, the ethics is involved there, that most of the time people say that anyway so many stem cells will be formed, it would be good if we can use them. There are people who say that even the embryo, we are killing it. If we are just removing some of the embryonic cells and just trashing others, so we are actually killing a human being. So that is where people who oppose stem cell research, they talk about. There are a lot of ethical considerations for stem cell research. There are safety concerns. So it is not only the ethics but even safety. For example, I gave you the example that people can use these embryonic stem cell for therapeutic, but because these embryonic stem cells, they are highly totipotent, so the chances of getting converted into some other form. For example, there is one of the very important thing is that many of these embryonic stem cells, because they grow very fast, so many of them can even develop into tumor, because property of a tumor is fast growth. And many of them, stem cell biologists says that there would be a lot of chances of immune reactions, immune rejections, which people of stem cell. This is just science based, but ethics wise, yes, safety and ethics is also very important. There are many misconceptions. People say that we just kill people, people, fetus will be killed, but there is most of the time the misconception is that even the people, they get the medically termination of pregnancy, they go for abortion, and then the blastocyst or stem cells are formed. No, most of the time we get the stem cells from two very common sources. One, the way I told you, that somatic cell versus germ cell electroporation. Second is in vitro fertilization labs are there, where if a couple wants to go for in vitro fertilization, at a time many embryos are they are fertilized, but implantation happens only for few. Only 2 or 3 implantations happen. So that means those leftover embryos can be used for stem cells. So there is never that for stem cell research, any kind of pregnancy is terminated or there is no abortion involved. Most of the time, it's stem cells which we are using, or stem cell, most common use of stem cell is they are coming from the in vitro fertilization labs only. Okay, so there are many. It's a good thing also that when you talk about, there are many cases. For example, diabetes. Treatment for diabetes, people are actually working where regenerative medicine and stem cell therapy has been proposed, where we talk about beta cells of the pancreas. They are the one which are not able to produce, which get destroyed during diabetes 1. So the stem cell research is being carried out where the B cells can be implanted. So pig beta cells are being implanted in the that work was done already, and there are cases where people are very hopeful that we will be able to regenerate beta cells of the pancreas. So these are some of the very important things which, although we have ethics involved, but yes, there are many cases with many studies which are which, again, most important ethical issues which is involved in human cloning and why it is not allowed is the misconceptions and the fraud. So there are so many cases, so many fraud, which this was the case in 2011 when people said that human cells are almost cloned. But there is one case of Huang from Korea. He actually committed suicide later. So there are many people who have actually gone up to a case where, and many of these papers have been retracted because the data was not reproduced after their group. Nobody could reproduce this data. So that's why it's not only ethical, but even the scientifically also it is not possible. So it is not only ethics which is involved for stem cell research and cloning, but even the scientific data is also supports that it's not easy to clone human. It's not very simple that you just, the way I was showing you this first picture, where you're just bringing two types of cells and just making an embryo and transplanting or placing it in the foster mother. No, it is not that easy. Because if you want to make an artificial environment, you have to give so many cytokines, growth promoting factors, exactly what is there in the natural condition. And it's not easy, you know. So that's why the chances of getting errors and many laboratory technical issues are very high. So that's why many of these papers which were published in PNS, one of the best paper, one of the best journal, even many of these, even Nature papers have been retracted. So if you see, whenever you talk about the fraud in human or medical sciences in the research, human cloning is one of the most common where you will see many cases of fraud and many cases where the papers have been retracted because non-reproducibility or things like that. And this is very. This is one person which I would like to share that although we are talking about a lot of cloning work, but still the in vivo work which is done in mice, there are some good reports also where people have used cardiac cells, because you know that brain and cardiomyocytes, these are some cells which once they are dead they cannot be repaired. Okay, because they are not dividing cells. So there are some very promising results also where people in vivo, in the animal models have got very good results, but they are not reproducible. They are not, when they go to human, it's not easy to get them the same result. So there are cases where this is one paper where people have actually, they could convert the cardiac fibroblast were very similar to what they see in mammalian heart. But once they start going and working, they start working on the mammalian heart, their characters. So it's not easy to do the same thing in a mammalian system. So this was something about the stem cell research and just. So you guys want to ask something about your exam and your till what we have done till now? You can ask, please. Yes, बच्चों. Guys, सब कुछ समझ आ गया? You people are great. Okay. Okay, guys, are you there? बच्चों, guys, बच्चों कहाँ
Yes,
हैं?
ma'am.
Attendance please. सो जाते हो तुम लोग, सो गए हो या क्या करते हो? हाँ, you just turn on your camera and go away. I mean, just log in for the class and go away somewhere. Okay. Anyway, um, attendance please. Mehak,
Present, ma'am.
Mihir,
Present, ma'am.
Ritu,
Present, ma'am.
Harita,
Present, ma'am.
Anvesha, Atul. Anvesha is absent. Atul, Sarang, Ritu Parna.
Present, ma'am.
Oh, Ritu Singh is different and Ritu Parna is different, right?
Yes, ma'am. Sorry, I mistook it.
You said attendance for Ritu Singh. Okay. Ritu
Yes, ma'am.
Parna.
Sorry.
Arpit,
Present, ma'am.
Madhurima, okay. Okay, guys. It's only 6 are present, huh? Okay. Madhurima absent, Sarang absent, Anvesha absent, Ritu Singh absent. Chali. Okay, guys. Bye-bye. So, next Friday, you guys, 4th, we have exam. You know that, right?
Yes,
4th
ma'am.
April. And it will be physical mode in 201, huh?
Yes, ma'am.
Okay? Chalo. Rest I will share information on the your WhatsApp group. Okay?
Thank you, ma'am.
Bye bye.